Good morning. Today we're going to study infection. What is infection? Well, basically infection is the invasion of a host organism body tissue by a disease-causing agent, their multiplication, and their reaction of host tissues to these organisms. Also, the toxins could produce an infectious disease. Infectious diseases are basically known as transmissible diseases or communicable diseases that they could either have a clinical evidence illness, such as signs and symptoms, resulting in an infection or not. Infections are caused by several different type of agents, such as viruses, bacteria, parasites, fungi, and even infestation with mites, fleas, lice, etc. Today we're going to concentrate basically on the different type of infectious process by bacteria, by viruses, by parasites, and by fungi. This is no more to illustrate that the body has a symbiotic relationship with ourselves. We do have bacteria and other type of microorganisms in our body at different levels that will protect you against infection. That is called normal flora. And what is normal flora? Basically, it's the aggregate of microorganisms that reside under either a surface or the deep layers of organs, the skin, the mucous membrane, and even our own secretions such as saliva, the urine, the feces, etc. It could include the same as the infectious organisms. It could include bacteria, fungi, archaea, etc. So this is widely known as normal flora or microflora. For example, bacteria population that could protect us, and they basically have the habitat on the skin. Could be the E. coli, staphylococci, streptococci. Basically what they do is that they have carbohydrate activate enzymes that basically colonize each site, and they're available to break down the sugars, and this is how they protect us from getting an infection. In addition, to mention some other examples, we do have actinomyces at the level of, for example, the mouth, to prevent tooth decay. In the vaginal flora, there's the lactobacillus that we explain on innate immunity. So if you think carefully, this type of normal flora, it belongs to your innate immunity that has a long life memory and presency, and only once the normal flora or the innate immunity is broken is when the second and the third immunity takes place. If you want examples of fungal flora, there's certain yeasts that occupy the human gut, such as Candida, and only when patients are immunocompromised, they develop the systemic fungal infection. Others in the skin could be actinobacteria. In the conjunctiva, we also have staphylococci, epididymitis, and aureus. We have in the lacrimal glands, we have lysosomal enzymes that are not aggressive. And those protect us against, for example, immunates to acquire Neisseria gonorrhea and Chlamydia trachomatis as a form of inborn conjunctivitis. So infection goes through different stages. There's a stage of colonization that basically um, you have an infectious microorganism that exists in any reservoir, for example, water, animals, or human to humans. Infections are contracted by this type of reservoirs, which could be animal reservoirs, mechanical vectors, such as the mosquitoes or the house flies, um, biological vectors, such as all these bacteria, viruses, um, areas of contamination, lice, um, also contaminated materials, such as fecal oral in the presence of either food poisoning or hepatitis A, D, and E, and also human to human. That's how we obtain infectious through droplets, such as coughing and sneezing, um, such as, for example, influenza or streptococci, pharyngitis, or bacterial meningitis. It could be physical contact, such as through blood transfusion or sex. In addition, you could obtain an infection from mother to child, which is called vertical transmission, either during pregnancy or through the birth canal, or to human, which is basically called horizontal transmission. So this way of obtaining infection is called colonization. Invasion is no more than the actual infectious agent that invades the local tissue, or it would spread through surrounding or distant tissue. Multiplication is the ability that the microorganism has to undergo a rapid multiplication through mitosis or through the process of transcription and translocation genetically. Spread is once the microorganism has multiplied and invaded the tissue, it would either be excreted um, through the urine, through the feces, through the bile, or through the pulmonary tract or the skin. So by any source, and basically that microorganism, after rapid multiplication, is able to either invade distant areas, invade the um, adaptive immune system through cellular or through humoral immunity, or to pass it on to another host. Now, they do this by a mechanism of either direct damage to the tissue locally through cellular metabolism, because the virus or bacteria or any type of microorganism could invade once they enter the lymphatics, the blood, or the internal organs. By either providing a cell dysfunction, if, for example, if they attack the nuclei of the cell and provide DNA synthesis to be impaired, or by toxic production, because many organisms will produce either an exogen pyrogen or endogen pyrogen. That basically what they do is stimulate the hypothalamus and stimulate the second line of defense, which is the inflammatory process, to start releasing interleukins, interferon, tumor necrosis factors. And in addition, all these cytokines will raise the thermoregulatory set point and provide fever, such as tumor necrosis factor, high catabolic state, and cachexia. So this is another way of spreading and contaminating the host until the cell is able to be dysfunctional or dead. So let's review some of the concepts. What is infectivity? It's the ability to invade and multiply of that particular microorganism. What is pathogenicity? It's the ability to produce a disease, because maybe the microorganism could have the ability to invade and multiply, but our adaptive immune system is strong enough and will impede the actual disease process to take place or the host to have symptoms. What is virulence? Is the potency that is required to kill the host. So it could be infective, it could be pathogenic, but if that particular organism does not go into a critical stage or the compensatory mechanisms never failed and they do not go into, for example, an infectious process that is able to go through sepsis and septic shock, but if the host is able to fight that infection because of a strong adaptive immunity and does not die, that particular organism does not have virulency. What is immunogenicity? It's the ability to produce an immune response. So again, you could be a there's a microorganism that has the ability of infectivity, but never triggers, even though it would break the innate immunity, the first line of defense, it would never trigger or activate the second line of defense, which will start at the level of the inflammatory cascade. What is toxigenicity? It's the degree of virulency, the degree of the potency that is required to kill the host. Now we will discuss the clinical infectious disease stages and port of entry and exit of the disease as well. What is port of entry? Is basically the route that the microorganism has chosen to infest the host. This is just to illustrate the host interaction with the microorganisms. Basically, they get in contact with the microorganism by any of the exposed area or port of entry. And if that microorganism is able to activate any line of defense, breaking first the first line and activating the second line, which will start at the level of the inflammatory cascade, is going to be able to produce invasion. If invasion occurs, then the microorganism will become established and they will grow. If they're able to produce an invasion, then infection will occur and of course the disease. Why? Because it will be a disruption of the tissue and in addition immunity as well. So once the host gets into the disease process, the morbidity and mortality percentage will be elevated. Now this is to illustrate again the port of entry, the host defense barriers, which is no more than your immune response, and how once a disease process will start, how the microorganism, besides choosing a port of entry, it would also choose a port of exit to continue spreading the disease. For example, it could be just an insect bite, or if we're talking about hepatitis, for example, if it's an hepatitis A, the port of entry would be the mouth. Then it would choose the target organ as the liver, and the port of exit would be the feces. And that's how the microorganism have chosen to spread the disease. I'm now planning you to memorize all these type of viruses, bacteria, and fungi, etc. It's just to give you an idea. What is, for example, a bacteria? A pathogenic bacteria is basically a bacteria that is able to cause an infection. Almost all bacteria are harmless because the majority belong to our normal flora. So it's basically beneficial to have them. But when the first line of defense has been jeopardized or broken, those bacteria could become pathogenic. Once the bacterial disease have chosen to burden um, the host, then a disease process will start. For example, pathogenic bacteria will contribute to many important diseases such as pneumonia, and those are, for example, streptococci pneumonia, pseudomonas, etc. They could produce foodborne illnesses such as Shigella, Campylobacter, Salmonella. There are many bacteria by location, for example. If you know that a patient comes in with a bacterial vaginosis, depending on the signs and symptoms and the quality of the secretions eliminated through the vagina, then you're able to do a differential diagnosis, which it would be helpful for health assessment between candidiasis or trichomonas. If the patient come in with signs and symptoms of headache, nuchal rigidity, fever, or altered mental status or seizures, you could be able to do a differential diagnosis among Neisseria meningitidis or streptococci or influenza, etc. For example, if the patient comes in with bacterial pneumonia, with signs and symptoms of a possible pneumonia, due to the actual review of systems and history of the present illness that you will learn in health assessment, you will be able to differentiate, as well as results in the laboratory and diagnostic test, if this is the potency viral versus bacterial infection, etc. If we're talking about viruses, well, let's talk first about different type of bacteria. When you go to pharmacology, we already discussed in the cell that the cell is surrounded by a membrane, and that membrane is called lipid bilayer membrane. But there is also an additional name that is called peptidoglycan, and that's no more than the bacterial cell wall itself. When you advance to pharmacology, you will be able to see that bacteria which are gram-positive, they have a different characteristic of the peptidoglycan. The layer for the gram-positive bacteria is thicker compared to the gram-negative, and has more permeability and is more lipid-rich. So for gram-positive bacteria, the antibiotic inhibition or the mechanism of action for antibiotics will be more enhanced for gram-positive bacteria than for gram-negative. Talking about viruses, what's the virus? Well, basically is no more than a disease process that are composed by an infection agent, which is a virus. It's a small organism that is able to attack living cells, and basically has a genetic material made from DNA and RNA as well. And there are multiple. You have norovirus or rotavirus, retrovirus, which we'll discuss today, the example of HIV. There are hepatitis as well as retrovirus, which are the viral hepatitis. There are multiple. So if we're going to continue discussing them. For example, for herpes virus, you have hepatitis E as well as herpes virus. You have the varicella, you have the actual herpes simple. Again, there are multiples. For example, adenovirus transmission is through droplet, fecal oral is the chosen area of entry and exit. It could produce adenovirus like an acute febrile pharyngitis. Coxsackie, for example, it could give you the hand, foot, and mouth disease. And the chosen exit and entry could be fecal oral and droplet as well. A very common one is Epstein-Barr virus, which is a type of herpes virus. The port of entry and exit is the saliva. That's why it's called the kiss disease. And it causes infectious mononucleosis. In addition, this is not the only disease that they contribute to. They also participate in Burkitt lymphoma and Hodgkin lymphoma. Hepatitis, how's the port of entry? Either body fluids or fecal oral or blood for hepatitis C and sexual contact, as well as B. So in advanced classes, in other semesters, we're going to discuss one by one, and port of entry, signs and symptoms, how is it diagnosed, and the treatment as well. Now, the infection could be spread as well through surface receptors. So the pathogens, they choose different type of mechanisms to invade the host and to also evade the host, to prevent the actual host or adaptive immune system or second line of defense to take over and kill them. So how do they manage to have a defense mechanism to avoid being killed by the host and take an opportunity to produce a disease? Well, they could do this by a surface receptor. Basically, they will have a unique receptor at the level of the cell that they could be transported into the blood and cause bacteremia. They could have antigenic variation. For example, the HIV virus is able to mutate, and that's how they would avoid being killed and provide pathogenicity to the host. They could also go through recombination, gene switching, production of toxins such as, again, exotoxins and endotoxins. Exotoxins and endotoxins basically are proteins released during bacterial growth. They're usually enzymes that have highly specific effects. They could produce a damage to any of the organelles, mitochondria, ribosomes, so ATP production is not going to happen, protein synthesis is not going to happen. They could also produce an antigenic drift, which is basically the production of new strains, or a shift, which is the recombination of new genomes. They could do gene switching, so basically they change genes to avoid immune response. That's how they go through rapid division and intracellular survival inside the host. Antigenic variation, again, basically is when they alter the surface molecule, or they break down molecules of the immune system through the presence of plasmin. They could evade the complement cascade from being activated. Or they could produce bacterial superantigens. Remember in the prior class, we discussed what a superantigen was, which is basically a toxin that is secreted during the bacterial growth that is able to produce an exaggerated response and either avoid or stimulate the histocompatibility complex, which is the antigen presenting cells. So we discussed a little bit already what bacterial virulence and infectivity is. They could have cytospore, which is iron receptors, to provide more virulency and infectivity. The bacteria, when we go through infectious disease in either family or acute care, and also in pharmacology, you would also see that the bacteria gram-positive have a thicker membrane layer, which is called peptidoglycan, than the gram-negative. We already discussed what's an exotoxin and endotoxin. It's basically those toxins that have the ability of lysing and providing bacterial growth. Now, this type of bacteria are able to be released and produce an endotoxic shock. Why? Because, again, they could be activated and provide a bacterial superantigenicity, an exaggerated response that would increase capillary permeability and leakage, producing edema in the third space, therefore hypotension and shock. Most commonly is present in gram-negative bacteria. So those endotoxins are released by vasoactive peptides, producing vasodilation, producing reduced blood pressure, decreased oxygen delivery, and therefore cardiovascular collapse. These endotoxins as well could activate the coagulation cascade, leading to an exhaustion of the fibrin degradation products, or D-dimer, and instead of having thrombotic events, patients would have massive bleed and end up in disseminated intravascular coagulation. This is to illustrate sepsis. Sepsis, as any other type of infectious process, once the first line of response or defense has been jeopardized, remember that the mastocytes will degranulate and histamine will be released. Interleukin will be released due to a damage of the endothelium. And that will produce what? Vasodilation, increased capillary permeability and edema. And at the same time, diapedesis, or the ability of the macrophages and the WBCs to adhere to the area of injury and provide more chemotaxis, or calling the other cells of the immune system for help. So after that, who's going to follow? Well, all the complement cascade will be activated. And after the complement cascade being activated, that they all basically merge at the level of the complement factor 5. The neutrophils will continue to present the antigen to the host and to the immune system, and that will produce more inflammation. Tumor necrosis factor gets activated, and remember, tumor necrosis factor, besides producing vasodilation, capillary permeability, and more calling of the macrophages, or diapedesis, or chemotaxis, which is the same thing. In addition to that, they will produce pain, they will produce more inflammation, they will produce cachexia, because the cell will go into a catalytic state or catabolic state. Macrophages, in addition, will release reactive oxygen system production, which is no more than free radicals, and that is toxic for the cell. that the cell could collapse and go into shock, and lysosomes could be released, and there is a lot of digestion in place, phagocytosis, and apoptosis, early apoptosis, pathological apoptosis of the cell. So this major vasodilation and capillary permeability is what contributes to the patient with sepsis to go into third space edema, ARDS, or adult respiratory syndrome. In addition to that, the complement cascade is not only activated, but also the coagulation cascade, having thrombotic events, consumption of the fibrin degradation products, and finally, DIC and death. This is another way to illustrate. This is the case of interaction of either gram-positive bacilli or gram-negative bacilli. Again, there is damage of the endothelium. There is release of multiple inflammatory mediators, macrophages, WBCs, going to the area of infection first, and continue chemotaxis of other inflammatory cytokines. But continues the cycle, activating more macrophages. Leukotrienes are released, prostaglandin are released, free radicals are released as well. The cellular immunity could be activated for the production of antibodies, or the T or humoral could also be activated, secreting CD4s and CD8s. I apologize about the lack of battery of the computer, but we're continuing the PowerPoint. So viral infections, they could do this by attachment, penetration, budding. Viral infections are, as we discussed already, they're most of the time they're self-limited. They do not produce endotoxins or exotoxins. They basically inhibit the DNA or the RNA, and they go through the same process of bacterial infections to stimulating the immune response, producing either hypersensitivity reaction, autoimmunity, and apoptosis at the end, exactly the same as bacterial infection. Fungi, they're basically called mycosis. It's the fungal infection, very common in the environment and under physiological conditions that could contribute to the development of spores. They could colonize the skin. They could produce persistent infections in the lung, in the skin, etc. The majority of the humans that are at risk for fungal infections is because they're either immunocompromised, or they're taking antibiotic, or there's a prior damage of a bacteria to the endothelium or to the mucosa, the mouth, the vagina, etc. So basically they have a weakened immune system. So if it's an elderly population, it's completely normal because in the elderly population, the immune system is decreased. But any patient that you see that is younger than 50 years old and they come in with a fungal infection, you have to investigate possible causes of immunosuppression, such as HIV has to be ruled out, cancer, And after that, you could, of course, question the patient, have you been on steroid treatment, any type of diabetes, have you taken antibiotic for a long time? Um, Mycosis could be superficial, such as tinea versicolor that you would see in skin disorders in your independent study. Um, could be cutaneous, such as dermatophytes that you're all aware of, tinea, which is ringworms. There's also systemic type of mycosis, which could be systemic candidiasis or cryptococci or aspergillus. How do you basically prevent skin fungal infections? Is to have a good hygiene. Skin should be clean and dry to prevent tinea versicolor. Um, and the treatment is, well, antifungal medication that you would see it on um, pharmacology. So what are the signs and symptoms of infections that infection could cause? Basically, malaise, weakness, very general signs and symptoms. Fever could be present due to the pyrogens, the toxins that we discussed, due to the exogens or endogens, pyrogens, or also to the tumor necrosis factor that produces fever and pain. Vaccinations could be provided to some to particular microorganisms, and if you realized in this PowerPoint, the schedules were excluded. First, because they were expired, and second, because it's already attached in your Blackboard site for your reference. Once you go to pediatric or adult, those vaccination schedules will be explained in detail. Now, let's talk about some mechanisms that the bacteria could choose to have antibiotic resistance. Even though this is particular information for pharmacology, is to give you an idea. We already discussed the mechanisms that they have to avoid infestation. To avoid the immune system to attack them and destroy them without producing an infection process. Now let's talk about antibiotic resistance, because it's a very hot topic that everyone wants to use antibiotic for a viral infection that is self-limited. So the bacteria could do this by enzymatic inactivation. So if, for example, we know that the adaptive immune system has the ability to secrete lysosomes and macrophages. Well, let me inactivate those enzymatic activations in order to avoid me as a microorganism to go through phagocytosis. Efflux pump is the one that is mostly recognized. It's a mechanism responsible for moving compounds like neurotransmitters, toxic substances, antibiotics, etc. It has a function of energy-dependent mechanism or active transport. They're localized in the cytoplasm. They're active transporter, as we said. Basically, if you avoid as microorganism to be transported from the cytoplasm to other macromolecules or organelles to go through the process of elimination, then of course infection will happen. So I would stay in the cytoplasm and go through a process which is called aromaticity, and that would impact me from the antibiotic to penetrate into my membrane and be eliminated. So what I'm trying to say is that the efflux pump is a mechanism of transportation. If I, as a microorganism, will avoid the process of transportation or having genetic elements such as chromosomes or plasmids be transported, I would not be eliminated. There are many medications that would inhibit the efflux pump, and those could be verapamil, for example, or beta carotenes could inhibit the efflux. Now, before I end this PowerPoint, I would like to talk a little bit about HIV. It's part of the adaptive immune system PowerPoint, but I would like to elaborate more than what was discussed in class. So HIV is basically an infection of retroviruses. There's two types, HIV-1 and HIV-2, that basically destroy CD4 lymphocytes. Remember, that's the cellular immunity, impairing cell-mediated immunity, increasing the risk of infections, cancers. The initial infection may not cause a specific febrile disease. Remember that the subsequent manifestations related to immunodeficiency is proportional to the level of the CD4 lymphocytes. HIV can directly damage the brain, the gonads, the kidneys, the heart. It could cause cognitive impairment, hypogonadism, renal insufficiency, cardiomyopathies. The manifestations range from asymptomatic carriage to AIDS, full-blown, due to serious opportunistic infections or cancer when the CD4 count is less than 200. HIV infection can be diagnosed by antibody, nucleic acid, HIV RNA, or antigen like P24 testing. That's going to be part of your more advanced classes. Now, let's talk a little bit about the pathophysiology of HIV. HIV basically attaches to and penetrates the host T cells via CD4 molecules and the receptors. Is an RNA that have several HIV encoded enzymes, and those are released into the host cell. Viral replication requires the presence of an enzyme which is called reverse transcriptase, which it will reverse. Remember that normally RNA is formed from the DNA. Well, this enzyme would do the reverse. That's why it's called reversed. An RNA dependent will produce DNA polymerase that would copy HIV RNA, producing proviral DNA. This copy mechanisms is prone to errors, resulting in frequent mutations. And remember, that's one of the mechanisms that microorganisms choose to avoid being excreted or eliminated from the host and continue infectious process. These mutations will facilitate the generation of HIV that can resist control by the host immune system and by antiretroviral drugs as well. This proviral DNA enters the host cell's nucleus and is integrated by the integrase enzyme into the host DNA in a process that involves, again, the integrase, which is another HIV enzyme. With each cell division, the integrated proviral DNA is duplicated along with the host DNA. Subsequently, the proviral HIV DNA can be transcribed to HIV RNA and translated to HIV proteins, such as the envelope glycoprotein 41 and 120. These HIV proteins are assembled into HIV virions, and the host cell's inner membrane embodied from the cell surface within an envelope of modified human cell membrane. Each host cell may produce thousands of variants. After budding, budding, protease, which is another enzyme, cleavage, viral protein, converting that immature virus into a mature infectious virus. So after this, all the infected CD4 lymphocytes will produce 98% or more of plasma HIV new virus. So a subset of infected CD4 lymphocytes constitutes a reservoir of HIV that can reactivate the actual disease process to continue. So two main consequences of HIV infection: damage to the immune system, of course, through CD4. CD4 lymphocytes are, remember, that are present in cell-mediated immunity. Their direct cytotoxic effect of HIV replication, because CD8 is also compromised. All the tissues that could be jeopardized is the dendritic cells present in the cell brains. That's why they have cognitive impairment and possibility of having any type of neurological manifestation and meningitis, tumors, etc. As the disease progresses, antibiotics to HIV are usually measured within the few weeks after primary infection. However, antibodies cannot fully control HIV infection because mutated forms of HIV that are not controlled by the patient's current anti-HIV antibodies are generated. So there are multiple copies of virus being produced, and that is also measured as part of diagnostic evaluations and treatment. Signs and symptoms: it could be from mild and non-specific infection, because there is an initial primary HIV infection that is asymptomatic, or could cause non-specific symptoms. Acute retroviral syndrome usually begins within 1 to 4 weeks of infection and could last up to 2 weeks. Symptoms could be mistaken by mononucleosis or because they're non-specific, such as fever, fatigue, any type of arthralgia, dermatitis. After the first symptoms disappear, most patients, even without treatment, have no symptoms or only a few mild or intermittent. Symptoms during the relative asymptomatic period could be, again, patients sometimes have lymphadenopathies that are confused with leukemia and lymphomas or any type of myeloproliferative syndrome. Oral candidiasis, remember, that's the reason why any young patient less than 50 years old, I would say 60 as well, less than 60 years old with any type of fungal infection should be investigated for HIV and cancer. Patients could present with herpes zoster. This is another disease that if you have a patient younger than 60 years old with herpes zosters, HIV or cancer must be ruled out. Because if it's after 60 years old, it's expected that the immune system for elderly population is decreased, so that could be normal, a normal finding. Patients could come in with diarrhea, profuse diarrhea, fatigue, fever, etc. Or sweating, night sweats. Now, when the CD4 count drops to less than 200, there are many clinical symptoms that could appear, such as Mycobacterium, pneumocystic Giroveni, cryptococci, fungi, etc. And as the CD4 declines, there are many other disease processes that appear as well. Cancer is another opportunistic infection, sarcoma, Kaposi. Hematological disorders are also associated with HIV, such as lymphoma and other cancers. Mycobacterial tuberculosis. Neurological syndromes could also be present as well, such as encephalitis, myelitis, progressive encephalitis, multifocal radiculopathies, meningitis, retinitis. Cardiac-wise, cardiomyopathies could be present due to HIV, renal insufficiency and nephrotic syndrome, esophagitis and gastritis, hepatocellular damage at the level of the liver, skiing, herpes zoster, simple. Pulmonary-wise, patients could have subacute pneumonia, not only due to pneumocystic uroveni, but also from cryptococci, neoformans, and histoplasma capsulatum. Diagnosis is basically by HIV test. You could do also HIV RNA, viral load, and CD4 counts and CD8s. It's basically done by ELISA and proved by Western blot. Treatment is going to be discussed in pharmacology. It's basically targeting all those enzymes that we discussed in the pathophysiology, the reverse transcriptase inhibitor, the integrase, etc. So thank you. This is the end of the PowerPoint. And I apologize for the battery loss.